It's been, I'm having a little trouble with this. It's been quite exciting. We've been all over the place, as you'll see. We've been in nice warm places with the gloves off. It's quiet. I'm down in Australia where we do a lot of our work. And we've been in places where you wish you had more than your gloves on. And that's the northern tip of Baffin Island in September. And I'm going to tell you that that was chilly. So we've started this massive project called ALGA, the Alga Life Global Audit. We started it in August 2009, and we have generated barcode records for 24,000 collections. And so this is a heat map of all the collections that we have barcoded and where they're from. And you can see that we have a lot from Canada, which is an area in which we do a lot of work, and we have a lot from Australia, which is kind of my second home, and is an area in which we do a lot of work. And most of these other collections, except for Africa, are also collections that I have personally made or have been done by members of my lab. So needless to say, the divorce lawyer is definitely getting itchy. The time away from home has been immense. Some years I'm away 4 or 5 months of the year on these trips. And people think it's just a vacation, but it's a heck of a lot of work. Because we've collected over 30,000 collections in the last 5 years and process them in these sorts of ways and manage to get the DNA sequence out of 24,000 of them. So it's been a huge undertaking, a heck of a lot of fun. But you know, when you're sitting there identifying an alga and it's 12:30 at night and you've already been going for 20 hours, you start to get a little grumpy. So there's a method to this madness. We literally go all over the world. We try to collect between 10 and 20 samples for each species, or what we think is a species. And we really try to broaden that out. So we try to get all the different morphologies that a species might have. We try to get all the habitats, sheltered, exposed, subtidal, intertidal. We try to get the full geographical range of every species. And you start to think about it, there's a lot of ocean on the planet, there's a lot of coastline. And even in Canada, we have our 3 different oceans to deal with, and we actually have the longest coastline in the world. So just the work in Canada can be overwhelming in its own right. But then when we do what we call molecular assisted alpha taxonomy, we do everything backwards. We sequence first, we lump all the things that have the same sequence together, and then we go back after the fact and we try to identify them in the more traditional ways. So we go about this sort of backwards from the traditional botanical approach. In addition to the fun of collecting the sample, all of this information has to be collected. So we have to get the species name, figure out what it is, classify it. We have to put a voucher collection somewhere. All of ours end up in the University of New Brunswick. We have to take an image of every collection that we do. We have to have detailed collection data for every image, lat, long, where it's from, right down to low intertidal on rock. And so there's a heck of a lot of databasing that goes on in the field for every record. And then when we get back to the lab, we have to generate this DNA sequence, and this all goes into the database as a record. So every collection has all of this information associated with it. So it's a very powerful database that we're generating. So what have we learned from 24,000 seaweed DNA barcodes? It's actually kind of scary. We have hundreds of new species, and I'll show you an example of the red crust in a minute. Even in Canada with 12,000 barcode records, we still find new species. Here's a nice new Phycodries from Meadow Cove just down the road that we collected in 2006. We have 1 collection of it. That's it. Hopefully we didn't make it go extinct. I'm sure it's out there somewhere. We're finding new genera, we're finding new families, we're finding new orders of algae. We have found 2 new orders of algae in this project. An order is something like the primates. That includes the humans, all the apes, all the monkeys, and the lemurs. We're finding completely new orders of life. This has really been an exciting time for us. And we're turning up all kinds of bizarre and interesting patterns along the way. And that's kind of the fun part of all this. So here's our example with the red crust. How weak is our current knowledge? We do a lot of work on these little red crusts. The postdoc in my lab, Kyle Dixon, who's sitting in front here, he's from Australia and he's working on these Paisonelias. We have about 1,400 collections of Paisonelia type things. Most of them are from Canada and Australia. We've got over 250 species, and there are only 70 species recognized on the planet. So we're already at 2.5 the number recognized, and we've really only worked in 2 parts of the world. There's probably going to be on the order of 400 species of Paisonelia within dust cells. And the way we estimate those things is we do these species discovery curves. So these these are the number of collections, and these are the number of genetic species we're hitting. And as long as these curves keep going up, that means we have to keep sampling. There's more species, there's more species, there's more species. When they start to level out and plateau off, that tells us that we've probably gotten most of the species in that group. We're getting into that sort of 95% range. And so these curves are going up, 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 up for the Pacenelias. There's another red algal cross-tilled Brandia. We have 350 collections, mostly Canada and Australia. We have over 50 species. There's only 16 recognized on the planet. So that's, I'll do some quick math, 4.5 times, something like that. And these curves are again going up. So there's a huge amount of overlooked diversity on the planet, species that we do not recognize yet that are out there masquerading as other things or looking as other things. Just quickly for the Bay of Fundy, there's about 100 species recognized. It's going to keep doing that. There are 2 Hildebrandias, we have 6, 2 Cetosiphons, we have found 4. Do I need that? I probably don't. 1 Pileella, we have 3, 2 Ectocarpus, we have 4. So I expect we'll increase our knowledge of the Bay of Fundy flora by about a 50% factor there. So there's lots of overlooked diversity even in our waters here in the Bay of Fundy. This is really important. A lot of times people don't understand why this might matter. But we have people trying to do biogeography and they don't even know what species are out there. We have people doing ecological and monitoring projects and they don't even know what species they're listing in their list of species that they found on the beach. This is my favorite one. There are people who do bioprospecting. They go out and they look for biochemical products that are going to be of use in medicine and things like that. And I've been at many talks with the guys that, oh, I went out and I collected Plocamium cartilaginum and I found a really good antiviral product. And then I went out the next day and I brought some plants in and they didn't have it, so we gave up on it. But we now know that Plocamium cartilaginum is actually about 16 different species. And so if you go out on one day and get one species and go out on another day and get a different species, you're not going to find that compound and you're going to give up on something that might be a very, very useful compound. So this is a serious problem in the bioprospecting business. The other problem is the ability to recognize introduced species. And I'll get to some examples of that as I work my way through this. So here's just a classic example of where things go horribly wrong. We have this Atlantic species called Porphyra linearis. Hannah Kucera worked on this in my lab. Very, very easy to identify, very distinctive. Late autumn, spring carpet on exposed rocks. It just coats these rocks, places like Peggy's Cove and whatnot. Easy to identify with its distinctive morphology, these very narrow, long blades. You can't go wrong. So, of course, we had to go out and collect a bunch of plants. We did about 72 collections from Rhode Island to Nova Scotia, extracted the DNA, and did the kind of things that we do. And it turns out that there are actually 3 species instead of 1 species that we are calling Porphyra linearis. In fact, true Porphyra linearis is extremely rare in our area. Only 2 of our 72 plants were true Porphyra linearis. Most of them are actually Porphyra umbilicalis, which a lot of people find hard to believe, because this is what Porphyra umbilicalis looks like. Unfortunately, when it gets into exposed habitats, it looks like this. And that's gone completely missed in the literature. And so here we have an example of convergence. Here we have an example of phenotypic plasticity. These 2 are the same species, these 2 are different species. And then we found this completely new thing here, which we call Pyropia peggycovensis, because we only get it at Peggy's Cove. It's rare, we only have 5 collections. It's actually the most linear of all 3 of them, despite this one being called linearis, and it's not even in the same genus. These are in the genus Porphyra, this is in the genus Pyropia, and they all look alike. So we couldn't even get them in the right genus, let alone the right species. An example with the kelps. This is work that Dan McDivitt did in my lab. There are basically only 2 species of Laminariaceae recognized in our part of the Atlantic. There's Laminaria digitated, which is very digitate, the blade is split, and Saccharina latissima, which is not. But of course we see plants like this, and you're like, well, is that, or is it just that that's kind of torn up a bit? And this is what got us looking at this problem and trying to figure out how many species of Laminariaceae were really in our area. Turns out there are 3 instead of 2. So there was the Laminaria digitata, which we would expect to see. There was the Saccharina latissima, which we would expect to see. And then there's this thing called Saccharina groenlandica, which is only known from the Pacific, despite its name, Groenlandica, which would suggest it's from Greenland. And we find it here in the Atlantic. This is the problem. This is Laminaria digitata. This is Saccharina latissima. This is Saccharina groenlandica. It can look digitate, just like Laminaria digitata, or it can have a plain blade, just like Saccharina latissima. So this one's masquerading as two different species in the local water. And so we're not just calling it one thing wrong, we're calling it two things wrong. And we've completely missed the fact that it's here. Now these things, fortunately, we can actually tell apart with microscopy. We get these things called mucilage ducts in kelp, which literally run snot up and down the plant. That's why they're so gooey. Saccharina latissima has none. Saccharina groenlandica has them in both the blade and the stipe. And in Laminaria digitata, you only get them in the blade. So there are some anatomical differences when you get in and look at these plants that match the genetic data, but you don't get to see them at the morphological level. We have an identity crisis in the seaweed world. We can't tell what things are. And I get this comment from so many people. If we cannot tell them apart in the field, does it make sense to recognize distinct species? And this question drives me nuts. Morphological convergence is very common. Cryptic is not the same as sibling or incipient. These things are sometimes in different genera. You can't just ignore the fact that they're out there because they look alike. It's ridiculous. The reason it's ridiculous is because the world isn't completely a human world. So I always challenge my students with this question. What would taxonomy be like if humans lacked sight? If we weren't using morphology for taxonomy in the first place, nobody would bring up the original question. And it's important to remember that the invertebrates and other organisms that are out there, depending on the seaweeds, sense the world very differently than we do. They don't key them out with a guide and a microscope. They have their own way of sensing the biochemistry of these things, and that biochemistry is very different. Here's an example, Caroline, who's also here, is doing this work, of where we can go wrong with this sort of stuff. So all the ecology that's been done on kelp in our area has been done on Saccharina latissima and Laminaria digitata. No one's recognized that Groenlandica is here. And so we can ask ourselves, does the presence of Groenlandica actually impact our ecological knowledge of these various species? So what Carolyn's been doing is going out and looking at these as 4 different categories. We have Saccharina latissima, we have the non-digitate Groenlandica, we have the digitate Groenlandica, and we have Laminaria digitata. And we can see there's a lot of latissima at sheltered sites and not so much at exposed sites, and that's all interesting, and that's what we believe to be true. What's interesting is there's very, very little of the non-digitate Groenlandica here. And so what we know about Saccharina latissima is probably true because the percentage of plants that we're getting mixed up with it is very, very small. You see exactly the opposite problem with the digitate plants. Here's the Laminaria digitata at exposed sites. This is the digitate Saccharina groenlandica. So right now, everything we know about Laminaria digitata is a combination of these two things. In fact, what we know about Laminaria digitata is probably more accurately what we know about Saccharina groenlandica, because there's more of it out there. But the problem is we have this composite knowledge. These two things have been lumped. And everything we know about Laminaria digitata is actually based on this composite of these two different species. And so what's true of species A may not be true of species B. Right now, Laminaria digitata is considered to be a perennial. Maybe it's not. Maybe Groenlandica is a perennial and digitata is only an annual. We have no idea of the true ecology of these species. And that's a problem.